paper entitled Inhibition of Herpes Simplex Virus 1 Gene Expression by Design Using Finger Transcription Factor. HSV1, or known as Herpes Simplex Virus 1, is actually a human pathogen that can cause mild epithelial lesion in human cell, and if it enters the nerve cell, it can become latent. Currently, most of the infection are being treated with acyclovir. However, some of the virus can mutate and hence become resistant to the drug, and this enables them to replicate in our cell. Therefore, this paper will evaluate the design of zinc finger transcription factor, which has a good potential to become a new antiviral treatment for HSV1 infection. Why is it possible? It is possible because zinc finger has the ability to bind to a wide range of DNA sequences, and 3 and 6 finger domain have been constructed and successfully activated and repressed single gene target, for instance, HIV promoter. HSV1 gene expression, basically it can be divided into 3 main stages, which are intermediate early, and late. And the IE genes and its product, IE175K, and the IE proteins are important in HSV1 gene regulation. In the promoter region of the IE gene, it contains at least 2 conserved enhancer sequences, TAAT, GARAT, and note that R is either nucleotide A or G. This sequence is recognized by the multi-protein trans-activation complex, which includes VP16. This complex will bind to the sequence, and once it binds to the sequence, it will activate the IE gene. Based on this whole concept, several zinc finger repressor proteins have been designed so that it will bind to the conserved enhancer sequence and inhibit the viral replicative cycle at the level of transcription. Why do we choose IE genes? Basically, there are a few reasons. First of all, the IE gene is expressed at the beginning of viral cycle. Hence, by inhibiting the expression of this IE gene, it means that we are inhibiting the infection at the earliest possible step. And secondly, the IE gene is easy to express. It's easy to access. The promoter region is easy to access, hence making it the best other target for the zinc finger to bind to. And finally, to investigate the competition between natural activator VP16 and artificially generated zinc finger for the same DNA binding site. They started the experiment by designing 3 finger proteins that can bind to 2 sequences. One is the conserved enhancer sequences, while the other is the sequence that is adjacent to it, and both of the sequences are located within the same promoter